to begin this unit, I'd like to review a concept that I've already used a little bit, so hopefully it hasn't thrown you too much. It's something you're probably already familiar with, but let's just make sure. So Bilateria, which is the largest, most diverse group of organisms on Earth, includes all bilaterally symmetrical animals. So that includes all of the deuterostomes, all of the protostomes. Hopefully that kind of is making sense to you. You're remembering back to our talking about animal diversity in some of those groups. So within the deuterostomes, we have the chordates, we have the echinoderms, right? So those are all primarily deuterostomes, some other smaller groups. And then within the protostomes, we have most of the different kinds of worms. We've got arthropods, we've got mollusks. So all of these organisms come from this bilaterally symmetrical body plan. Now, there have been some revisions to it, and we'll talk about some of those groups when we look at the individual body plans of some of those other types of animals. But for the vast, vast majority of all animals on Earth, they follow this body plan. And essentially, the way it works is there are 3 relevant axes. And these axes sometimes have different names. These are the names we're going to be using. They're the most commonly applied. Sometimes if you're dissecting the planes across which you will dissect, like transverse, sagittal, things like that, there are different terms that are used, but we're not going to worry about these. These are the most commonly used ones and the ones that are most critical for us. So the first one is the anterior to posterior axis. So the anterior end, of course, is the head, and the posterior end is the tail. Now realize that for some organisms, depending on their orientation, like humans are a good example, if our orientation has changed relative to our ancestors, there might be a little bit of difference in where we draw the anterior to posterior line because we're trying to draw it so it's the same in all organisms. So head to tail, basically, right? So we wouldn't do head to the bottom of feet because humans have this modified bipedal gait, which is very, very different from our ancestral condition. So we still do the same one from head to tail, even though humans have a tiny little short right coccyx at the end. But that's still our same anterior to posterior end. So we want all of these, when we draw them, to be the same basic axis, no matter how much they've changed and modified. And for most organisms, it's pretty straightforward. Okay, so we're using this fish as an example, but we could put an insect here. We could put a cow, a dolphin, any organism would fit in here, and we could do these same axes. The second major axis is dorsal to ventral. This one's usually pretty easy for most people because dorsal fin's a fairly common term. So if you want to think of it as back to belly or back to front, although again, front can be a little bit problematic of a term because as organisms' orientation to their environment changes, what sometimes people mean by front is different from one animal to the next. So dorsal, the back, ventral, the stomach side maybe is the easiest way to think of it. And again, for most organisms, it's pretty straightforward. They maintain this very simple ancestral form. Now, humans realize we keep our dorsal side now because we stand vertically. Our dorsal side is still our back side, but it's no longer pointing up when we're moving around, unless we're crawling like a baby. But just because our orientation changes relative to the environment doesn't mean that these basic fundamental axes of our bodies have changed. So we just need to make sure that's clear. So dorsal, ventral, back to stomach. And then the third one, sometimes people use the term lateral, and they've got it here, lateral, left and right. This one we're going to use for our developmental purposes. We're not going to worry about left to right. Excuse me. And the reason why we don't use the terms left and right, or they're not as important developmentally, is because in bilaterally symmetrical animals, the left and right are essentially the same, particularly externally. Sometimes internally there's some slight differences like lobes of the liver, hearts, moved over to one side a little bit, but for the most part, our left side and our right side are identical. So rather than defining left and right, we will instead look at proximal to distal. And this can refer, it's primarily looking at this third axis away from, if you want to think of it like the x-axis is the anterior to posterior, the y-axis is the dorsal to ventral, and then we'd have this third axis that goes perpendicular to both of those, the z-axis. And we use proximal, meaning close to the body, and distal, meaning far away. So if you put your arms out, and this is really useful for appendages and appendage development, if you stick your arm out, the proximal side would be where it attaches at the shoulder, and then the distal side would be the very tips where your fingers are. And then sometimes people talk about medial, kind of intermediate areas of that. Excuse me. But we'll just basically do the main ends of the axes. So proximal and distal. So you should recognize those three main. Now, humans like to look for patterns. We like to look for things that make sense in our head. And sometimes we see patterns where there's really not any deeper meaning. And that can be a little bit of a problem. So the question when people first began to explore development, and most of this work took place first in Drosophila, they were looking for genetic pathways that corresponded with these three main axes that were obvious and you could see in pretty much all bilateria, right? So they're saying, okay, at a molecular level, at a developmental level, what genes are patterning these axes? And it turns out that there are groups of genes, and many of these are very, very conserved across all animals, but there are groups of genes that are responsible for patterning these three major axes. Now, we're not going to do all three of them. We'll do proximal to distal, and we've kind of already looked at that a little bit, right? When we looked at the chicken wing and that zone of polarizing activity, and even more importantly, that apical ectodermal ridge, which helped to pattern the proximal to distal axis of the developing vertebrate limb. But we are going to focus primarily, although we may come back from time to time and give examples of proximal to distal, and maybe even just refer very briefly to dorsal to ventral. We're going to primarily focus on the anterior to posterior patterning and what goes on there, because that was the first to be explored. It's one of the most important, fundamental to all bilaterian animals. Okay, so let's look at this and how it works in the fruit fly. And it's actually a really good example of evolution and changes and how we can get stability of form despite some of the underlying genetic mechanisms changing. And we'll talk about that as we explore more with the fruit fly. So we're going to come back to this first maternal gene that we already mentioned, bicoid, right? So many of these early genes, the maternal genes that are first put down in the egg by the mother, are very, very fundamental patterns. They just establish this kind of very broad difference between one end of the developing embryo and the other. And so for the AP axis, this anterior-posterior axis, one of the very, very key genes for this in the fruit fly is bicoid. Okay, so we already mentioned that. So bicoid, among a small handful of other genes, is responsible for helping to establish the initial anterior to posterior axis of the developing fruit fly embryo. And all maternal genes are like that. They just do very simple differences. And then they target different genes based on threshold, based on diffusion of these patterning genes through the tissues. They target different genes. Those genes then target different genes, and it gets a little bit more complex at each step. And so what we are going to do for this anterior to posterior pattern, it's still fairly simplified, but we're going to take you through the first five major steps. And at each level, it's going to get a little bit more complex. So review if you have a bicoid mutant, if you mutate, knock out bicoid so it's not expressed, or alternatively, if you were to ablate, right, cut off this little bit of the anterior end of the embryo at an early stage, that signal would no longer be there. You'd have no signal saying, hey, this is the head is, and the recruitment of all of the other genes that then build the head are just, that is absent. And so we end up with a mutant with basically a headless fruit fly. Now, obviously, you have to assess these at a very early stage because without a head, they can't feed, they can't move correctly, coordinate movements, and they just lay there and eventually die. Although they are born with living cells, and you can verify that phenotype. Okay, so the very earliest genes after this anterior to posterior axis. So first step A, we're going to call it, are these maternal effect genes like bicoid that do front to back. The next step are something called gap genes. And gap genes begin to outline the basic idea that this organism is going to be segmented. Now, segmentation is a common feature for all bilateria, but some of them it's a lot more obvious, particularly external, right? So you look at a human, and externally we don't look very segmented. Although internally there are still large hints, leftover indicators of our ancestral segmentation, right? So look at our vertebrae, which are set up in segments. Many muscle groups, like abdominal muscles, are these repeated patterns that are still left over from this ancestral segmented. But in fruit flies, it's easy, right? You can see the segments externally. They correspond to repeated units internally, and so the segment is a basic module of all animals at one level or another, but especially still very, very fundamental in arthropods like the fruit fly. And so gap genes are the first hint of this segment formation. So gap genes, and there are a number of them. We won't talk about all of them or name them, and I just want you to know the general term gap genes. So maternal genes impact gap genes. The gap genes set up the basic segment formation. And so if we mutate different gap genes, we're going to end up with deformities in the formation of these segments. So this diagram is a fluorescently labeled markers of each of the segments. In red, we have the thoracic segments, and one of the gap genes being expressed to outline those segment patterns. And then the green ones are for the abdominal segments. And so notice that if we mutate these gap genes, we don't get any thoracic segment formation. So all the red markers are gone. And we have some major deformities in the abdominal segment formation. So these are critical, and they're turned on by the maternal genes, and they're critical for proper formation of the segments. Okay? The third step is what we call the pair rule genes. And the pair rule genes begin, in a very broad sense, to differentiate segments one from the other. So that segment A1 is going to be different from segment A2 and A3, and so we have different genes then being expressed in these different segments that allow them to go on to a different fundamental fate. Okay, and so many of these are kind of either even or odds. So this is one called Fushitarazu. Don't need to know that name, but it is a pair rule gene, and if we turn off its expression, we lose all of, I guess it'd be the even segments, right? Although the numbers start over here, but so we lose T2, we would lose A1, which is 2 down from the second thoracic one, and so we lose every other segment. Okay, so again, review. Maternal genes establish the very basic anterior to posterior pattern. The gap genes do initial segmentation development. The pair rule genes then start to establish basic differences between segments. And so, for instance, some of them are in charge for every other segment. The next major group of genes, which we're going to do as D, are segment polarity genes. So these help to define what the front part of the segment is going to be and the back part of the segment. So now we have segments established. We have basic segments next to each other being slightly different. And now within each segment, we're going to establish a secondary anterior to posterior pattern, right? So we've got the complete anterior to posterior signal, which was partially established by bicoid. And now within that big field, we have secondary fields that also need to be established. The axes of those need to be established. And so secondary anterior to posterior axis is established within each segment, saying, hey, this is the front of the segment where this gene is expressed. And then it will diffuse back to the back where there's a much lower signal representing the back of that segment. Right? So front to back of each segment. And so if we mutate these, we lose that clear definition of front to back, and we end up kind of blurring all of the segments together. So you can see the clear, distinct difference in when we mutated this. It's a gene called gooseberry. The gene name is derived because the mutant version of it looks kind of round and over like a gooseberry without clear segments. I don't even know if you know what a gooseberry is, but that's what the first researchers thought it looked like. So we lose good definition of front to back segments. Now, the very last, and there are more steps beyond this, especially as segmentation development gets more and more defined. But the last one that we're going to talk about in the basic anterior to posterior patterning of the fruit fly are the Hox genes. And the Hox genes are activated by these different combinations of all these 4 groups that have come before. So maternal genes come on and they turn on the gap genes. The gap genes then turn on the pair rule genes. The pair rule genes then turn on different segment polarity genes. And finally, those different combinations of genes will turn on different Hox genes. And Hox genes, at this point, we begin to identify and define the different regions of the body. So the thorax is different from the abdomen. And within the thorax, the first segment, second segment, and third segment are different and express different combinations of Hox genes from one another. And so we really have at this point pretty basic but still really nicely defined regions of the body. And so we can see this very clearly in the expression patterns of these Hox genes. And I've showed you this image before, right? And so there are a couple of trends that I just want to reinforce. Number one, there is a clear pattern from the anterior to the posterior. And remember, this is kind of unusual for developmental genes, but for the Hox genes, that pattern in which they are expressed in the organism from anterior, here's the head of the fruit fly going all the way back. It's kind of like in a fetal position, so it's folded over on itself. And there's the posterior end. So anterior folded around like a fetus all the way to the posterior. So we have this clear layering of genes from the anterior to the posterior axis. And remember that those are found in the genome in that same pattern. And so we call the entire group of Hox genes the Hox complex. And because that complex is in the genome in the same order that it's used in the developing embryo, we give that a name, and it's unusual. It's called collinearity. Again, remember, for most genes, their spatial orientation in the embryo really doesn't have anything to do with where they are found in the genome. But an exception to that rule are the Hox genes. Okay, so that's one of the trends that we observe is this collinearity. The other trend that we observe is this trend of overlapping expression, where we might have some areas where there's only labial being expressed in pink. And then there's a small area where both labial and deformed, the pink and the green, are both expressed. And sometimes it's hard to see with the colors they've chosen here, but you can see an area where both sex chromosomes reduced, SCR, and deformed, DFD, are expressed in the same cells. And so the nice thing about this is with 8 Hox genes, we can pattern very specifically way more than 8 segments, because we have some genes that have 1 Hox gene, some genes that have 2 Hox genes, and some genes that may even have 3 different Hox genes that are being expressed. So notice near the abdomen here we have a region where abdominal A, abdominal B, and ultrabithorax, this UBX in red, are all expressed at some levels. A much higher level of expression here in the first abdominal segment. Okay, so the 2 major trends that we see is in these Hox genes is the anterior to posterior matching with the genome, collinearity, if you just want to shorthand that, and overlap of genes to establish many differential segments. And so a group of cells that gets all 3 genes, like abdominal A, abdominal B, and UBX, is going to develop into reproductive structures, for instance. A gene that expresses antennapedia and UBX in the fruit fly is going to develop wings, but very, very modified wings into halteres. And so remember, when we turn off UBX and when we only have antennapedia present, that's when we get 4 wings, because the modification that is done by UBX is no longer present. And so we end up with more of those ancestral type wings. Okay, so we will use this anterior to posterior and these Hox genes as our key kind of core example throughout the class, and we'll talk in a lot more detail both in Drosophila and then a little bit in other organisms also. But remember that as complicated as this is, there are just as complicated patterns to pattern the dorsal to ventral axis. And we could look at genes and a number of genes that do that. And if we mutate those genes, we'll get differences in the dorsal to ventral patterns. Okay, and in the same respect, there are proximal to distal genes. And we're not going to look at any of those. The only ones I want you to know, and the ones we'll use as kind of our key placeholder, are these anterior to posterior genes and the network, or maybe you can, if you want to think of it more as a series, that's fine, but there's really feedback and other connections, so it is a little bit more network-like in how these genes interact to lead to this basic fundamental. And we'll talk about some of them in a lot more example for the next lecture, and in your reading for this week, we're going to focus on UBX and abdominal A and the role that they play in defining the insect as a 6-legged arthropod, rather than like the ancestor, which had legs on nearly every segment. So look forward to that. Do the readings for reading paper 5A and 5B. It's really a great example of a key innovation, a change that we can point at the genes and even the parts of the genes that changed to lead to this major revision in body types.